Okay. So you are responsible for understanding the path of blood through mammalian circulation. So where blood is entering and where it's exiting. And we're going to go through this, and also it's in your book, but you are required to understand the entire pathway. Okay. And this includes the systemic and pulmonary pathways. Okay, and we know by now that blue represents deoxygenated blood and red represents oxygenated blood. Okay, so
The heart is the pump of the human circulatory system. The left side of the heart has two connected chambers, the left atrium and the left ventricle. The right side of the heart also has two connected chambers, the right atrium and the right ventricle. These two sides, or pumps, of the heart are not directly connected with one another. Oxygenated blood from the lungs travels through large vessels called the pulmonary veins and enters the left side of the heart, emptying directly into the left atrium. The pulmonary veins are unusual in that they carry oxygenated blood. Other veins, because they carry blood back to the heart from the body tissues, carry deoxygenated blood. From the left atrium, blood flows through a one-way valve called the left atrioventricular valve, also known as the bicuspid valve, into the left ventricle. Most of this flow, roughly 70%, occurs while the heart is relaxed. The atrium then contracts, filling the remaining 30% of the ventricle with its blood. After a slight delay, the ventricle contracts. The contraction forces the blood to exit into an opening that leads to the largest artery in the body, the aorta. The atrioventricular valve closes and prevents the backflow of blood into the atrium. The aorta is closed off from the left ventricle by a one-way valve, the aortic semilunar valve. It is oriented to permit the flow of the blood out of the ventricle, but it snaps shut in response to backflow. Many arteries branch from the aorta, carrying oxygen-rich blood to all parts of the body. The pathway of blood vessels to the body regions and organs other than the lungs is called the systemic circulation. The systemic circulation brings blood to the neck and head and to organs in the rest of the body. The systemic circulation gives up oxygen to the body tissues and receives carbon dioxide. The blood that flows into the arterial system eventually returns to the heart after flowing through the capillaries. As it returns, blood passes through a series of veins, eventually entering the right side of the heart. Two large veins collect blood from the systemic circulation. The superior vena cava drains the upper body, and the inferior vena cava drains the lower body. These veins dump deoxygenated blood into the right atrium. Blood passes from the right atrium into the right ventricle through a one-way valve, the right atrioventricular valve, also known as the tricuspid valve. Blood passes out of the contracting right ventricle through a second valve, the pulmonary semilunar valve, into a single pulmonary artery, sometimes called the pulmonary trunk, which subsequently branches into arteries that carry deoxygenated blood to the lungs. The blood then returns from the lungs to the left side of the heart, replenished with oxygen and cleared of much of its load of carbon dioxide. The pumping of the heart is a repeated cardiac cycle of relaxation and contraction of the atria and ventricles.
okay, so you have four valves in the heart. Okay, these are flaps of connective tissue and they prevent backflow. So the first is the atrioventricular valve, and this is between the atrium and the ventricle. This keeps blood from flowing back into the atria when the ventricles contract. And this is the lub sound, the first sound that you hear in your heartbeat. Okay? And then semilunar valves. Okay, these are between the ventricle and the arteries. And this is the dub sound. Okay, they prevent backflow from arteries into ventricles while they are relaxing. And there are pulmonary and aortic semilunar valves. Okay. Okay, so the closing of valves, the lub is the recoil of blood against those closed valves. And the dub, which is the second sound, the lub dub, is the recoil of blood against semilunar valves. Okay, and so if you have a heart murmur, this is a defect in your valves that causes a hissing sound because there's a stream of blood that's squirting backwards through the valve. Okay, so reading a cardiac cycle graph. So when you're looking at the bottom here, you can see your heart sound, which is the first, the lub, the second, dub, and then the third, which usually you can't hear, right? But still exist. And this is all about blood rushing against those valves when they close. Okay, so the first, that lub, is the blood that is rushing against that atrial ventricular valve as it's closing. Okay, and then again, the dub is when the semilunar valve is closing. Okay. So one complete sequence of pumping is the heart contracting and pumping, the heart relaxes, and the chambers fill. Okay, so the contraction phase, so this is systole. Okay, this is ventricles that are pumping blood out. And then the relaxation phase is the dystole phase, and this is where the atria is refilling with blood. Okay, so systolic over dystolic is the peak pressure over minimum pressure. Okay, and this is how your blood pressure is calculated. And it's calculated in millimeters mercury. Okay, so measurement of blood. Okay, high blood pressure or hypertension, if your top number, which remember is your systolic, is greater than 150, and if your bottom number is greater than 90. Okay, so remember it's a measurement of that peak pressure over the minimum pressure. Okay, so the heart does not receive an impulse from your nervous system. Okay, it's myogenic, meaning that it has its own built-in neurons. And this is a sinoatrial node, the atrioventricular node, and this has to do with the Purkinje tissues. I can never say this correctly. So this is about the electricity of your heart, okay? And this comes from pacemaker cells that are in your sinoatrial. And we're going to watch an.
The human heart has 4 chambers. The 2 upper chambers are the atria, and the 2 lower chambers are the ventricles. The interatrial septum separates the 2 atria, and the interventricular septum is the partition between the 2 ventricles. The right atrium and right ventricle are connected through a tricuspid valve. As the name suggests, it has 3 leaflets or cusps. On the other side, the left atrium and ventricle are connected through a bicuspid valve. The bicuspid valve is also called the mitral valve because of its resemblance to a bishop's 2-sided mitre or hat. The leaflets of these atrioventricular valves are connected to fibrous tissue called the chordae tendineae, which in turn are attached to papillary muscles. Contraction and relaxation of these muscles make the valves open and close. There are also valves at the opening of the pulmonary trunk and the aorta. These are called the pulmonary valve and the aortic valve, respectively. Because of their crescent moon shape, these 2 valves are also called semilunar valves. The characteristic double-up sound of the heartbeat is produced during the closing of the heart valves. The thickness of the walls of the 4 heart chambers varies with their functions. The walls of the atria are thinner than those of the ventricles, as the blood needs to be pumped into adjacent ventricles only. The left ventricle pumps blood a greater distance at higher pressure. Therefore, the wall of the left ventricle is thicker than that of the right ventricle. The sinoatrial node, atrioventricular node, bundle of HIS and Purkinje fibers represent the specialized cardiac tissue. These fibers are autoexcitable as they have the potential to generate electrical activity without any external stimuli. This makes the heart beat continuously.
So cardiac muscle differs from the muscle in other areas of the body, like you just saw, like we talked about a second ago, because it is myogenic. So this means that it naturally contracts and relaxes. It does not need to receive impulses from a nerve to make it contract. In fact, if cardiac muscle cells were cultured in a warm, oxygenated solution with nutrients and whatnot, they contract and relax on their own. But individual heart and muscle cells cannot be allowed to contract on their own because they would have their own natural rhythms. And if they did, they wouldn't contract together and it wouldn't act like a pump. The heart has its own built-in controlling and coordinating system, which prevents this from happening. The cardiac cycle is initiated in a specialized patch of muscles in the wall of the right atrium called the sinoatrial node, and it's often called SAN for short, or pacemaker cells. And you can see here in the steps here on this model, number 1 is the sinoatrial or pacemaker cells generates the impulse, and each time that the muscles of the SAN contract, they send a wave of electrical activity, which spreads out rapidly over all of the atrial walls. And then the muscle in those walls responds by contracting, and at the same rhythm as the SAN is putting forth with its electrical signal. So all the muscle in both the atria contracts almost simultaneously. And we know by the lub-dub, right, that the muscles of the ventricles do not contract until after the muscles in the atria. So this delay is caused by a feature of the heart that briefly delays the excitation wave as it passes from the atria into the ventricles. So there's this band of fibers between the atria and the ventricles, which does not conduct the excitation wave. So really, it's a non-conductor. Okay. So as the wave spreads out from the SAN over the atrial walls, it cannot pass into the ventricle walls because of those fibers that are non-conducting. So the only route then is through this patch of conducting fibers situated in the septum, which is the middle part of the heart here, via a patch of those fibers called AVN, okay, and that's the atrioventricular node. Okay, and you can see these right here. Okay. So the AVN, the atrioventricular node or bundle as it is in this model, picks up the excitation wave as it spreads across the atrial walls, but it is at a very slight delay and passes it on to a bunch of connecting fibers called the, I always say this wrong, Purkinje fibers. And this runs down the septum between the ventricles. So this will transmit the wave to the base of the septum where it spreads outwards and upwards through the ventricle walls. So again, it starts in that sinoatrial node. It moves through all of the atrium walls, essentially at the same time they contract, but we don't want the atrium and the ventricle contracting at the same time. Otherwise, all blood would be constantly pushed in and then pulled back into the heart at the same time, which we don't want. So because of that, there has to be this non-conducting tissue that will slow that wave so that the ventricles will contract after the atrium, the walls of the atrium do. So the cardiac muscle will contract from the bottom up, so squeezing blood upwards and into the arteries. So I encourage you to use my links here and watch these animations. Okay, so an electrocardiogram or an ECG, this can be used to detect and record the waves of excitation flowing through heart muscle. Electrodes are generally placed on the skin over opposite sides of the heart, and then the electrical potential is generated with time or recorded over time. And so the result is essentially a graph of a voltage against time, which you see here on this guy's shirt or right here, the pink line that's above, and this is what we would see. This is the ECG. This is the line we would see at, at a hospital when they're measuring our heart rate. And you don't really need to know much about electrocardiograms, but it is pretty interesting to know what you're looking at, especially now that you kind of understand how electricity is moving through the heart. So the part that's labeled P here represents the wave of excitation sweeping over the atrial walls. So that's that first that's moving from the sinus node down. And then the Q here, R and S represent the wave of excitation in the ventricle walls. And then the T section indicates the recovery of the ventricle walls. Okay, so now you kind of understand what you're looking at. So if you follow this link here, there's a website for diagrams and visuals that can help you understand that too. Okay, so we've talked about hemoglobin. We talked about it in chapter 2 when we talked about biological molecules, but I'm just going to remind you a little bit about what exactly hemoglobin is. So hemoglobin is a protein, okay, that has an iron or a heme group, okay, so that heme group has an iron atom in there, and it binds to oxygen, okay, and hemoglobin is needed to store and carry oxygen, but it also has to be able to release it from the blood at the right time to the cells that need it, right? Because those red blood cells carry oxygen through the hemoglobin protein all throughout our body, but they need to be able to drop it off when it's necessary. So how does hemoglobin know when to bind to oxygen and when to release it? So the answer is that hemoglobin is constructed in such a way that it takes cues from the conditions and concentrations of oxygen and carbon dioxide around it. So hemoglobin is a tetramer. It's composed of nearly 4 identical subunits, each of which contains that iron atom in that specific heme group. And it's part of the hemoglobin protein that can bind to the oxygen molecule. So each hemoglobin subunit is capable of changing its shape or conformation between 2 basic forms depending on a variety of factors. When oxygen binds to a heme group, the hemoglobin subunit will change its shape so that the second, third, and fourth heme groups can more easily bind with oxygen. So it's like once one oxygen binds, almost instantaneously the next 3 heme groups bind to an oxygen. Okay, and it's the ability of that protein to change shape. The process of changing shape is called cooperativity. Cooperativity. So, in an oxygen-rich environment, like the capillaries of the lungs, oxygen can be rapidly taken out of solution and stored in hemoglobin because of the cooperativity. Cooperativity. As blood enters capillaries and tissues that are actively using oxygen, the low concentration in the tissues draws dissolved oxygen out of the blood and into the tissues, leaving very little oxygen dissolved in the blood. So without a high concentration in the blood, oxygen bound to hemoglobin is free to go back into the solution in the blood. And so in addition, active tissues have a high concentration of carbon dioxide, right? Because those have been going through cellular respiration. So hemoglobin can also bind to carbon dioxide, so that oxygen gets released by the hemoglobin into the blood. So let me show you here. So this is called the hemoglobin dissociation curve. And the shape, okay, of this curve reflects the way that oxygen atoms combine with hemoglobin molecules. So up to an oxygen partial pressure of around 2 kilopascals, an average on average, only one oxygen molecule is combined with each hemoglobin molecule. But once this oxygen molecule is combined, it becomes successively easier for the second and third oxygen molecules to combine. So the curve rises very steeply. So over this part of the curve, a small change in the partial pressure of oxygen causes a very large change in the amount of oxygen which is carried by the hemoglobin. So this leads us to the idea of the Bohr effect. The Bohr effect, in general terms, is a change in oxygen affinity. So oxygen affinity means how much oxygen hemoglobin will bind with. Okay, so it's a change in the oxygen affinity of hemoglobin with a change in pH. And the effect is beneficial at the tissue level, where lower pH decreases oxygen affinity and promotes oxygen release. So what this is saying is that when the pH decreases, so in other words, when our blood becomes a bit more acidic, then that will trigger a chemical reaction which allows hemoglobin to release that oxygen. Okay, and in turn, it will pick up that CO2, and that's really incredible if you think about it. I mean, the whole idea is that hemoglobin is carrying oxygen around your body, but at some point, it has to be able to know, right, in quotation marks, when to release that oxygen. And it knows that when it gets into an environment that is a little more acidic, meaning that there's a little bit more CO2. And I'm going to show you that chemical reaction in a second, but there's more CO2 in that environment, meaning that more cells have been respiring and that there is a need for oxygen in that area. So then that CO2 will cause a chemical reaction within the hemoglobin, which allows it to drop off that oxygen and pick up that CO2, taking it out back to the lungs to be respired as gas. Okay, so the amount of oxygen that hemoglobin carries is affected not only by the partial pressure of oxygen. Remember that if we're in the lungs and it's a really high concentration of oxygen, then hemoglobin will really readily bind with that oxygen. It's also really affected by the partial pressure of carbon dioxide. So when there's a high amount of carbon dioxide and creating an acidic environment, then the blood will release that oxygen and bind with that CO2. Okay, so carbon dioxide is continuously produced by respiring cells. It diffuses from cells into the blood plasma, where it diffuses into the red blood cells. So in the cytoplasm of red blood cells, there's this enzyme that's called carbonic anhydrase that catalyzes this reaction. Okay, so hemoglobin readily combines with the hydrogen ions, forming hemoglobic acid. And in doing so, it releases the oxygen which it's been carrying. So the net result of this reaction is really twofold. The hemoglobin will bind with those excess hydrogen ions, which are formed when the carbon dioxide dissolves and dissociates. And so a high concentration of hydrogen ions would mean that the blood is being more acidic. But if the hemoglobin is picking those up, that is decreasing or increasing the pH of the blood. So by removing those hydrogen ions from the solution, hemoglobin is helping to maintain the pH. But also, the presence of high partial pressure of carbon dioxide causes hemoglobin to release that oxygen. And that is the Bohr effect. It is exactly what is needed. High concentrations of carbon dioxide are found in actively respiring tissues, which need oxygen. These high carbon dioxide concentrations cause hemoglobin to release its oxygen even more readily than it would otherwise do. Okay, and so that leads us to that dissociation curve. So if you're looking at the dissociation curve for hemoglobin at high partial pressure carbon dioxide, it looks like the lower curve that you see here. Here. Okay, so at each partial pressure of oxygen, the hemoglobin is less saturated than it would be at low partial pressure of carbon dioxide. So therefore the curve lies below and to the right of the normal curve. Okay, so you have a lot to study, and we will talk in class.